Hello, my name is Caden Kresak.
Hello, my name is Nick Cowan.
And my name is Chris Pierson. And we are presenting Beach Bums and Bacteria, examining the potential impacts of the Coastal Renourishment Project on the abundance of E. coli.
Our research question was, how does beach renourishment and their profiles impact the abundance of E. coli and sand grain size across different beach zones?
Erosion is a coastal threat to ecosystems and communities to nearly 70% of our coastlines worldwide. Beach renourishment is a common response and one we use here locally in St. Augustine. This local process costs $60 million and dredges sand miles offshore from pipelines to add sand to our beaches. In 2014, a study was begun in Folly Beach, South Carolina to examine how the renourishment may have impacted E. coli concentrations and their sand grain size, and if there is any correlation between. We decided to replicate this at our own beaches here at Anastasia.
Escherichia coli, more commonly known as E. coli, is a common indicator bacterium with hundreds of different strains. A further understanding of our local E. coli concentrations on the beach could further public health, safety, and kindle more testing for other harmful pathogens.
Our hypotheses are: 1. There is a significant difference between E. coli levels and nourishment status. 2. There is a significant difference between E. coli levels in the dune and intertidal zones. And 3. Sand grain size significantly affects E. coli levels.
So where are our sites? Our 4 sites are located on the north end of Anastasia Island. Our 2 renourished sites are at the Pier and A Street, where our non-renourished sites are south at F Street and Ocean Trace Road. We pile 3 deep up front in the truck and drive to Anastasia Island. We began gathering our tools and supplies to collect sand out at the beach. Rubber gloves, 8 sterile plastic bottles, some DI water, and a small spade shovel.
Standing in the middle of the beach at each site, one of us would throw the quadrat into the intertidal zone and the dune part of the beach to ensure randomization of sand collection.
Using the sterile shovel, we would scoop enough sand into the bottles for both sand sieving and E. coli testing. We disinfect the shovel with DI water to ensure that the sediment data doesn't get mixed with our other samples.
We immediately drive back to the lab where we would begin the testing process. Using a small sterile vial, we mix a spoonful of sand with distilled water.
Shaking the mixture for approximately 2 minutes, we used a pipette to place exactly 8 drops each into each sample onto the labeled Neogen test kit. We made sure to wear gloves to ensure proper lab safety and not to spread E. coli. After all 8 test kits were ready, we placed the kits into the incubator at about 35 degrees Celsius for about 24 hours.
We came back the next day to count the E. coli colonies. We then put the sand in the drying oven. Once dry, we took it out and began sieving.
When the sieving sand is dried in the oven, each sample must be filtered separately through the sieve stack. Because each sample varied in amount, we calculated percentages of silt and clay, very fine sand, medium sand, coarse sand, fine gravel, and gravel.
Now time for our results and discussion.